Thanks, David, for the invitation and also the introduction, and especially selecting Chinese songs and before the talk. And it's my great pleasure to be here. My talk today is about this neural network reconstruction and by using causal measures. And actually, the talk consists of serious works on this topic. And these are my collaborators, including some experiments who help to verify our methods. And basically, Yan Yang is now a faculty member at Shenzhen Institute of Advanced Technology, and he focused on these simulated neuronal networks. And later, Kai and Zhongqi extended the theoretical results to real data. So, okay, so we know that different species have different brain structure in terms of neural number, but the common feature is the brain actually span across multiple spatial scales from this nanometer synapse to millimeter neural circuits. And thanks to the development of the experimental tools, along with the computational power and artificial neural networks, we have now actually accumulated a large amounts of experimental phenomena and data for analysis. But however, the data analysis is often a problem in neuroscience, as this featured an issue by nature, and which often leads to the irreproducible experimental results. And the solution are suggested by Hinton and Sinofsky, meaning we have to do very quantitative math. And so my lab focus on the mesoscale of the brain, meaning starting from single neuron and to build neural networks. And we investigate the relationship among structure, dynamics, and function. And this includes the intersection between experimental neuroscience and applied math. So basically, if we treat the brain as a complex network, it can be started in 3 levels. So first is the network node for single neuron. So we have actually investigated how does a neuron integrate synaptic inputs, what's the quantitative algorithm, and how to correctly infer this synaptic conductance in experiment, and even how to build a precise and effective mapping between biological neuron and artificial neuron. The second is how this neuron connects to each other, namely the network structure. So we have, we know this seminar is dedicated to Carl van Vreeswijk, who did fundamental works on this balanced state. We also have studied the balanced state, but for this heterogeneous connectivity structure like scale-free, and also we have also investigated whether for different species, and the variant diagram have the common principle. And then the third is about the function, meaning once we have each node and we connect to form the neural networks, we can focus on certain brain region or even the large-scale whole brain to try to answer the encoding and decoding issues. So we have investigated the large computational modeling of early visual pathway and early olfactory pathway about how they encode the sensory inputs, what's the properties, how to map the input to the neural response, and what's the possible compressed coding principle. So basically, this just summarize of works in my labs. So today we focus on the second level, the network structure, basically how to infer the connectivity from the network's outputs. So before that, let's first look at how in experiment they obtain the connectivity. So we know the Human Connectome project is a data source, also a benchmark on this topic, but they focus on this macroscopic level, meaning the connectivity among brain regions are obtained by this diffusion tensor image. So for the cellular level, meaning connectivity among neurons, recent works for this mouse prefrontal cortex or even the fruit fly and also the monkey brain, they also have just tried to, through big data analysis, to obtain the connectivity among neurons. But overall, all these projects, takes large amounts of manpower and time. And meanwhile, we have also, developed many experimental tools to measure brain activity. So for the cellular level, we have 2-photon calcium image, and also we have neural pixels, which actually provides a stable and long-term neural activity recording and over large neural populations and this long time scales. So, and in addition, the measurement of brain activity takes less efforts than that of brain structural connectivity. So our question is whether it is possible to infer the structural connectivity from activity measurements. And actually on this topic, there have been some works. So for example, based on both signals, and one can calculate the correlation coefficient to obtain the functional connectivity, and then compared to the structural connectivity among brain regions obtained by diffusion tensor image. So the good news is these two connectivity are correlated. And however, which gives the possibility to infer the structural connectivity from functional connectivity. Meanwhile, recent works using an idealized ring model, they found that such inference may be related to the network dynamics. For example, when the recurrent strength is strong, and often the inference error is large. And the second is even the structural connectivity may not change, but different brain states may possess different functional connectivity. The third is the computation of functional connectivity is method dependent, meaning here not only just we can use this correlation coefficient, we can also use other statistical methods like Granger causality and like Bayesian or mutual information and so on. And often, different methods we compute and we obtain different functional connectivity. So all these just give challenges to infer the structural connectivity from functional connectivity. So our question is whether, for certain statistical method, we can actually quantitatively build a mapping between the statistical connectivity and the structural connectivity, thus to achieve the inference. So the causal measures I will introduce is called Granger causality. So suppose, let's say we have n time series. So what is the Granger causality connectivity or GC connectivity between the xi and xj? So here, for example, we draw this diagram. We want to characterize whether, xj is directly connected to xi or, it's have this indirect causal connectivity through some other nodes. So basically, the formulation is follows. Suppose we first can consider this conditional autoregression. So basically, let's say we want to predict the future of xi and use its own history and also the history information of all other nodes. And then we have the prediction error, or called the residual of the autoregression. And meanwhile, we can also consider the conditional joint regression, meaning here we predict the future of xi, not only just use its own history information and the history information of other nodes, but also we include the history information of the jth node. So we essentially obtain the joint regression residual. So suppose, let's say, by taking into account the history information of xj, and if this prediction error is less than the auto regression error, then we say including the history information of xj will help to predict the xi's future. Thus, we say there's a GC connectivity from xj to xi, which mathematically is defined as a log ratio of variance between this autoregression residual and the joint regression residual. And similarly, for the xj to xi can be also defined. So here, once we have this statistical method, we focus on two widely used models. One is Hodgkin-Huxley neural models, including ion channel dynamics. The other is the simplified version called this integrated phi model. I think most of our audience may be familiar with these two models. So the formulation is as follows. So suppose we have this HH and all these leaky integrated phi neuronal networks. Once we give the structural connectivity, then we can evolve the system and then obtain the voltage traces for each neuron. And based on these voltage traces, we can compute the Granger causality to obtain the this GC connectivity matrix. And then we compare to the original this structural connectivity matrix called the adjacency matrix. And what we found interesting is they are highly consistent with each other. Actually, we have tried also different connection number or different dynamic regime. So now the question is, this why for this nonlinear HH and leaky integrated phi neuronal networks, we can establish or we can obtain a one-to-one mapping between the structural connectivity and this GC connectivity. So to understand this, we need to try to use math to extract the underlying mechanism. So here, let's say if we pick up two nodes from a network, one is called xj, one is called xi, and from xj to xi, there's a unidirectional connection and with a coupling strength S. And remember this, when xj, it spikes, and often it will just send signals to xi and will induce the change of the state of xi. So intuitively, this can be characterized by the what's called this spike triggered response. So basically for each xj spike, and we just look at the xi's response within certain window after its spike, and we put it here. And also for the another xj spike, we look at the xi's response with the certain window after that and put it here. Then we consider the statistical average. So intuitively, we can think, so if xj is not connected to xi, then this statistical average normally gives this flat pattern. And however, if the xj is connected to xi, so which means whenever xj spikes, that should induce some change. So statistical average should give this non-flat pattern. So that's what's essentially obtained. Indeed, we have this xj connected to xi, and this spike trigger average, we obtain this non-flat pattern. However, on the other hand, the xi is not connected to xj. If we consider this spike triggered, the voltage, it also gives us the non-flat pattern. So this, so why this inconsistency happens, or why this is not consistent with our intuition? So the reason is actually for voltage time series, it has self-correlation. So intuitively, suppose let's say we have this time series y and time series x, And the true interaction is the y interact to x. And however, since the time series of y has its own correlation structure, so meaning its future will depend on its history. So this will lead to the spurious causal interaction from xn to y, the future of y. So meaning to exclude such spurious causal interaction. So the central part is we somehow need to remove the self-correlation. So after trying some time, what we found is actually, if we look at this autoregression, this formulation, and not just looking at the voltage time series. Instead, we look at the autoregression residuals because it's naturally the whitened signals. And indeed, we focus on these spike-triggered residuals. And for the spike-triggered residual under this J spike, we see this non-flat pattern. And this is consistent with the connection from xj to xi. On the other hand, for this I is not connected to xj, we look at the spike-triggered residual the other way around. It gives us the flat pattern. So this means instead of looking at the voltage, we should somehow focus on the autoregressing residuals. And so that's can give this consistent this connectivity feature. And also we can perform the asymptotics and to finally obtain that spike-triggered residuals actually is related to this correlation structure among this autoregression residuals. So basically for this whether under condition of J spike, we look at the I spike of the residual, and this is correlated to the correlation of the autoregression residual between I and J, with J is the history. And once we have this, and then we know that voltage and this residual actually have this inverse linear relationship, then we can also prove that if by looking at the GC connectivity between voltages is the same as the GC connectivity between residuals. And then we switch to the GC connectivity of voltage to the GC connectivity of residuals. And finally, we can obtain the GC connectivity is the summation of the square of this correlation coefficient. And here with xj to xi, that is just a correlation coefficient between this the i's residual and the j's residual, with the j becomes the history. The other way around is the I becomes the history. And finally, we can achieve the GC connectivity is proportional to the square of the coupling strength. And this just based on the asymptotic analysis. Now, next, we want to verify whether numerically that's true. And indeed, for a neural network, we checked different dynamic regime from this high fluctuating regime to this almost mean-driven regime. And this quadratic relation holds for different dynamic regime, which means now, indeed, we can, based on voltage time series, we can calculate its Granger causality connectivity and then use it to infer the structural connectivity. And we know what's the underlying mechanism. And then the next question is whether, this GC is for, for general nonlinear system, this also can help to just infer the structure connectivity. Then we tried for two this linear Gaussian variable, indeed, GC connectivity is consistent with the structural connectivity. However, if we just under very simple nonlinear transform, and we obtain the new time series xt and yt, and the true connectivity is still unidirectional. However, we compute the GC connectivity. That indicates there's no connectivity between x and y. And so that gives inconsistent result. Meanwhile, if we consider this time-delayed mutual information, which I will introduce later, basically it can be regarded as a nonlinear version of this correlation coefficient. And this gives the consistent connectivity with the structural connectivity. And so which means GC, for this nonlinear HH or leaky integrated file, we obtain the consistency of GC connectivity and the structure connectivity. However, for other general systems, it's easy to get these inconsistent results. And so actually, in previous works, they have proved that only for Gaussian random variables, this GC connectivity is actually the same as this connectivity characterized by this information theoretical matrix, like the transfer entropy. So that's why, in Nobel lecture, the Granger said, he actually proposed this Granger causality, and the definition was pragmatic, meaning once you have time series, you can compute the GC connectivity. But many ridiculous papers appeared. So if we look at the growth of this GC, this Granger causality, it actually increased. So from our works, basically, is we know that's for HH and this leaky integrated file works, we basically GC connectivity is consistent with the structural connectivity. However, for general nonlinear system, we should be very careful. Now we have questions, since we have many other causal measures. So what's the relation among different causal measures? And the second is for certain system, how we should choose proper causality measures to compute. And another thing is, so in previous works, we studied the HH or leaky integrated file models. We focus on voltage time series. But if we want to push the method to the real data, we know the subthreshold voltage normally is very noisy. Instead, we should focus on this high signal noise to ratio, like the spike train obtained through these neural pixels. And the second big issue, if we also want to extend our method to the real applications, is sometimes in experimental, we cannot measure or obtain the information of the whole network. Basically, we can measure part of the network, and we have this unmeasured neurons. So the question now becomes whether, only measure part of the neuron, we can cook up certain method to just obtain or infer the connectivity among this subnetwork. So basically, we need a pairwise reconstruction, meaning we don't need global information. We just need the partial information of the network, and we do the inference. So next, we focus on this pairwise network reconstruction. And first, we try to answer what's the relation among different causal measures. And the second is whether, these causal measures is consistent with structural connectivity. The third is we want to extend our method to real experimental data. So here, the causality measures, we focus on 4 widely used methods. This Granger causality I have introduced, and I will introduce later about this time delay mutual information. and also this time delay correlation coefficient and transfer entropy. And so the signal we focus on is this spike train signal may be obtained by neural pixels or even just inferred from this calcium image signals. And we try to infer the synaptic connectivity among neurons. And so first is what's the relation between different causality measures. So here they say the time delayed correlation coefficient is defined as the typical correlation, but just with parameter m, indicating the y is a history and x is a present. And the Granger causality we also adapted with the time delay that contains the parameter m, and the time delayed mutual information. I said it's actually a nonlinear version of this correlation coefficient. Instead of focus on the correlation structure, they focus on the whether two signals are independent or not. And this transfer entropy with time delay parameters and also are sort of different from the classical sense. Later, I will just show that's by the introduction of this time delay parameters helps to overcome the curse of dimensionality of this transfer entropy and also this Granger causality. And so if we plot a picture of this full method, so here the left is linear and the right is nonlinear. Basically, the GC and the TDCC are linear method, and the TE and the TDMI are nonlinear. And this bottom to up is whether you consider this history information or not. So the formulation is the following. Suppose, let's say, for an HH network, we evolve the system, and although we have the voltage time series, but we focus only on the spike trains. And for these spike trains, we binarized and we obtain these digital signals, like with each component is 0 or 1, indicating whether the neuron spike or not. And based on these binary signals, so I'll just ignore the details. Basically, we can obtain 4 algorithms, and we establish the mathematical relation between each this causal measures. And if we plot a diagram like this, so for each theorem connects 2 causal measures, and so basically these 4 can be just linked with this expression. And this theorem is proved under the assumption, let's say the neuron, the coupling strength is not too strong. And the second is the neuron has this refractory period, which is also consistent with the physiology of the neurons. And so especially I want to emphasize, for this pulse output signal, we obtain the consistent or the same relation as the previous work proved for this Gaussian random variable. So basically we can extend their results to this pulse output signals. And so once we have this pairwise, this causal measures, and we know what's the mathematical relation of them, the next question is whether we can use them to infer the structural connectivity. So this can be studied. Suppose, let's say, we have pick up any 2 nodes, and suppose that's a unidirectional connected. And for the pulse output, so we know that our causality measures have the parameter m indicating the delay, so how it is selected. So basically, for 2 time series, we compute the time delayed mutual information. We pick up the peak value of the TDMI, and it corresponds to certain delay time. And this is used as a time delay parameters. And so for the inference, we know that actually we have two distributions. One is called when two nodes are connected, we have this causal values distribution. The other is when two nodes are not connected, we have this distribution of causal values. So the good inference is, suppose these two distributions are well separated. Of course, we know that's we can achieve very high accuracy of reconstruction. However, if these two overlaps, so which will give us the bad reconstruction. And so here we tried that for 100 HH network with random connectivity, the percentage of connection is 25%. And we obtain, by evolving the system for each neuron is driven by this Poisson input. And we obtain the rest of this spike train. And we can just compute the distribution of causal values. Indeed, they're well separated. And so actually the big one corresponds to the directly connected pairs, and the smaller one corresponds to the pairs without direct connection. And so even though for this whole network, we just measure this 20 neurons, this subnetwork information, and use this information of subnetwork, we do this pairwise reconstruction. Still, we can obtain this well-separated two distributions. And so which means somehow, this pairwise causal measures is consistent with the underlying structural connectivity. And now the question is similar with what's the underlying mechanism. So here, similar to previous work, and we pick up two nodes, and suppose one is Y and one is X. It is unidirectional connected with coupling stress S. So in previous works, we focus on the spike-triggered response. Here, after trying some time, we found that we can actually introduce what's called the spike-induced change of firing probability, meaning by looking at the state of X when it is fired. So whether the Y with certain delay when it fires or it does not fire, the probability, the difference of the firing probability of Xn. And for this, we can obtain this TDCC, time delayed correlation coefficient, is proportional to this spike-induced change of firing probability. And also it is proportional to the coupling stress. And then finally, we can also obtain this quadratic relationship between these four causal measures and also the coupling strength. So that's essentially tells us why for two nodes are connected, you can use this pairwise, this causal measures to infer the connectivity. And but there's other question, right? So different from previous work, now we only have this partial information of the network. So basically we have some unmeasured nodes. So which means, so even though X is not directly connected Y, but it can just get this causal connection through some other nodes, or X, Y can have this common nodes. And since this nodes is not known, so Y, for this indirect, is causal connectivity will not influence our will not influence the inference of our network inference network structure. So to answer this, what we actually found is, so suppose, let's say we consider from Y to X is just the indirectly influenced Y to X through certain node W. And the from Y to W, they have this direct connection with coupling stress S1. From W to X, they have this connection stress S2. And so for in previous analysis, we know that for this direct connection, and we have this spike-induced change of firing probability is proportional to the connection stress. Now here we want to say this spike-induced causal this firing probability from Y to X, even though they are not directly connected, but actually they are proportional to this product of the coupling stress. And indeed, we can obtain such analytical relation. And note that in previous, we also obtained this T relation between TE and this delta P as that's quadratic relation. So we can obtain similarly for this causal measures of the transfer entropy have such relation, meaning from Y to X, even though it is not directly connected, but somehow the transfer entropy is proportional to the product of the two directly connected transfer entropy value. And note that the transfer entropy value is often small, so which means the indirect connection of the transfer entropy value is often much smaller than direct ones. So this means the direct, once the nodes have direct connection, the distribution of these causal values will be much larger than those unconnected one or even those indirect connected one. So indeed, we also tried this numerical simulation to verify our theoretical analysis, and so that's explains why this indirect connection will not influence this network inference. And so in previous work for simulated work, we always know the ground truth. Suppose we don't know ground truth, so then how to get the results? So basically, is if we don't know the results, but once we have this causal, the distribution of causal values, we can always just separate it as this summation of two distributions, and with larger one, we define it as the distribution of connected pairs, and with the smaller one, we define it as a distribution of the disconnected ones. And then we have four different parts. And so this FP and this Fn is false positive and false negative. And that's the inference error. And this green and the blue part and corresponds to the correct ones. And so quantitatively, this can be characterized as this AUC, area under the curve. So basically, suppose we have this distribution of causal values, we separate it as a summation of two ones, and then we can just choose different criteria, meaning the causal values larger than that we define as connected, smaller than that we define as unconnected. Then we just scan this criteria, we obtain a curve between this false positive and true negative. So then we have this area under the curve, and so basically it's close to one, meaning this distribution are well separated. The reconstruction is fine, and if these two overlaps a lot, this AUC is just will be 0.5. So basically, it's like a random guess. So indeed, we also tried a different connectivity structure, like this normal distribution, uniform distribution, exponential, and log normal. And we always get this high AUC number. And also for different neural models, we can also obtain this good network reconstruction. And so sometimes we know that the neurons may just have this moderate, this synchrony issue. So once we see this sort of this synchrony structure, we can just perform this downsampling. Basically, we remove those synchronized events, and for these downsampled rasters, we can just use them to do this network inference. Still, we achieve very good results, and this just for excitatory, and also for excitatory and inhibitory, we also can obtain the good network reconstruction. And even for this EI balanced network, we can also get good results. So finally, to the experimental data, what we tried is Allen Institute, and they measure the mouse visual cortex, and actually the drive is standard drifting, grating, or static grating, or even natural movie or natural scene. And so for each different drive, we compute this AUC is also very high, and also these 4 different causal measures, and they are very close, indicating our theoretical relation holds. And also for each pair of stimulation, we say, although the functional connectivity can be the same, but here the structural connectivity sort of fixed. So we can just compute whether, for different drive, they do inference, whether they are consistent, since the underlying structural connectivity, we think, do not change. So still we can achieve very high consistency. So, and also for the reasons, we collaborated with Lin's lab, and at ECNU, they study this different inhibitory neurons, how they contribute to the rhythmic of the local field potential. And use our method, we actually found a certain neuron called theta neuron, meaning they just participate in the theta rhythm of the local field potential. And by performing realistic neural simulation, we essentially found that the inhibitory neuron they measure is probably the CCK type and not the other type. So, and so finally, I want to say that's the limitation of our reconstruction. So we sort of need the relative long recording of the firing events. For example, this neural pixel data, we need this about 10 or 20 minutes long. And also we focus on this asynchrony or this only moderate synchrony for highly synchronized. So there's no way you can tell, the causal structure or the between neurons. And finally, we focus sort of like the sparse connectivity, because for very dense connectivity, so even though the indirect path, the causal connectivity is small, but you have many different indirect paths. So the accumulation of them still, may just become larger, and then this may give you the incorrect inference. So that's the summary of my talk. So basically, for conditional Granger causality based on multi-time series, we illustrated the mechanism, and we propose pairwise the reconstruction of network algorithm based on this pulse output data, and we establish the mathematical relation among different causal measures, and finally we can use our method to experimental data. So these are just related publications. For more information, you can just visit our lab website. So thank you.